Hello everyone. Today we will learn about process of recombinant DNA technology, one of the very important topic in our biotechnology discussion. DNA manipulation is basic to all biotechnological processes. The different methods employed to combine different genes of interest are generalized by the term called process of recombinant DNA technology. This is the most basic aspect we need to learn to explore the possibilities in biotechnology. And there are 6 stages in this process. First stage is isolation of genetic material. Second stage is restriction enzyme digestion. Third process is amplification using PCR. Fourth stage is ligation of DNA molecules. Sixth stage is insertion of rDNA into host, and sixth stage is isolation of recombinant cells. Let us learn each stage. Now let us study about first stage, that is isolation of DNA or genetic material. The first step in recombinant DNA technology is to isolate the desired DNA in its pure form. That is, it should be free from other macromolecules. To achieve that, we must follow this three-step process. That is, rupturing of all cell membranes to release the cellular components and DNA. The second step is separation of nucleic acids from other cellular components. And the third, purification of nucleic acids. All these procedures have been developed and standardized by understanding the physical, chemical, and biological nature of different types of organisms like bacteria, fungi, plants, and animals. General standard procedures to obtain pure DNA from these sources will involve first rupturing the cells and treating them with the enzymes like lysozyme or cellulose. Then the ruptured components predominantly containing proteins as debris will be denatured with the help of detergents like SDS, sodium dodecyl sulfate. Further, enzymatic treatments to remove other macromolecules will include proteases and RNAs to destroy proteins and RNAs. Centrifugation technique will be used to separate cell debris from the DNA containing supernatant. So supernatant, we will get the DNA, and such DNA will be precipitated with the help of ethanol, where we will get the viscous strands of DNA in a proper manner, and purified DNA looks like a white mass of the compounds, loop like structures. We can scoop it, and a lengthy loop will come out from the uh, tubes. Let us explore second step in the recombinant DNA technology, that is restriction enzyme digestion in detailed manner. Here, restriction enzymes act as a molecular scissors that cut DNA at specific locations, and these reactions are called restriction enzyme dimensions. So here you can see a restriction enzyme and here DNA molecule, and let us see what happens to them. So you can see that they have restriction enzyme has cut the DNA molecule at the specific site, and the two strands have been separated, and they involve the incubation of purified DNA with the selected restriction enzymes at conditions optimum for that specific enzymes. Later, the fragments of the DNA are separated by size using gel electrophoresis. A current, since DNA is negatively charged, a current causes the DNA fragments to move away from the negative electrode and towards the positive point. As the DNA travels through agarose, the larger fragments get stuck in the gel pores more than the smaller DNA fragments. So this is how we can separate different lengths of the DNAs which are cut by the restriction use of the restriction enzymes. Both vector DNA as well as the sample DNA can be processed in the same way. Third process of recombinant DNA technology is amplification of the selected DNA, that is desired gene, using PCR technology, polymerase chain reaction. Polymerase chain reaction, or PCR, is a method of making multiple copies of a DNA sequence using the enzyme DNA polymerase in vitro. It helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. PCR reactions are run on thermal cyclers. Equipments like this, thermal cyclers, using the particular components like templates, which is a small DNA template which is DNA to be amplified, with the primers, which are small chemically synthesized oligonucleotides that are complementary to a region of the DNA. Then we need enzymes, specifically Taq polymerase, DNA polymerase, and nucleotides which are needed to extend the primers by the enzyme. So this is how the multiple copies of the enzymes will be obtained by the PCR. And again, the whole animated version is picturized here. You can see that there are basically three processes are there in the polymerase chain reaction which takes place in vitro. First is denaturation, and it takes place in the temperature range between 94 to 96 degrees centigrade, then annealing at nearly 68 degrees, and elongation. And the same processes, denaturation refers to the double stranded DNA will become single stranded. Then what happens, annealing refers to the primers will go and attach to that particular molecule, oligonucleotides will attach to the DNA strand. Then elongation takes place in vitro. The same cycles will repeat, and within 6 to 7 cycles we can get thousands of copies of DNA which we wanted to work out further. Let us explore fourth stage in the recombinant DNA technology, that is ligation of DNA molecules, and it includes ligation is a process of joining the DNA with the help of a specific joining enzymes called as ligases, where the two ends of different or same, whatever it is, the DNA molecules, ultimately DNA, now we have arranged at the space that DNA is only thing, from where it is secondary, but depending on their nature of the restriction enzyme cuts, the ligation enzyme recombines the DNA, and this particular DNA is termed as recombinant DNA, and that is the ultimate function of ligases, the enzymes which are very much required for the purpose of recombinant DNA. And here basically what happens, the DNA fragments of the vectors and DNA fragments from the gene of interest, both will get hybridized, or they will be ligated, they will be glued together to form hybrid DNA. The fifth stage in recombinant DNA technology is insertion of recombinant DNA into the host, and that step number 5, insertion of recombinant DNA into host, is done with the process called transformation. Basically what happens here, usually the bacteria are not willing to give entry to any kind of other foreign particles within them. So we have to make them to get this particular DNA, that is, which is recombined DNA, from the external source into the bacterial cells for a multiplication purpose. So what we will be doing, there are so many processes. So usually the shock treatment will be given, and electric current will be given, temperature treatments will be given, or different types of other treatments will be given, and by through those treatments what happens, the bacterial cell wall will become imbibable to the plasmid, and once the plasmid, the recombined plasmid, the recombined vector or vector carrying recombinant DNA gets into the bacterial cell, then it can cultured based on the its resistance selected marker components, and usually antibiotic resistance will be considered as the selected marker, and the hosts which are having the antibiotic resistance will be grown in the specific medium, and which are not, they will die off. The last but not the least, the sixth very important stage is isolation of recombinant cells. So the cells which are grown in the after insertion of the modified DNA, they grow in specific other cells, and we have to isolate only recombined cells to get our desired product, and that can be done. Again, continued transformation process generates a mixed population of transformed, here you can observe transformed and non-transformed forms. I have depicted this particular thing in this manner. This is purely for understanding purpose. It is not the all these things will happen within a organism, but conceptual understanding I have used these graphics. So selection process involves filtering the transformed host cells only, and for isolation of the recombinant cell, a selectable marker will be used, and once the selectable marker uses, that means selectable marker may be antibiotic resistant gene. What happens? The only by the use of the selectable marker genes, only the recombined or which are having the property to overcome that particular thing will resistance will be grown, and they are termed as transformed hosts. So this is how a recombinant DNA technology processes can be summarized. At the end of the isolation, we can use, by the use of the bioreactors, we can grow them in the multiple downstreaming process which is called as, and we can get the product of our interest. So by learning these 6 stages, that is the isolation of genetic material, restriction enzyme digestion, amplification using PCR, ligation of DNA molecules, insertion of rDNA into host, isolation of recombinant cells, we can perform function of recombining the DNA into another organism for our benefit. Hope you like this particular lecture, and thank you for listening.